Okay, and it appears that we are officially live. So hello, everybody, and welcome back to another session of our Sussex Vision Seminar Series, as always within the Worldwide Neuro Initiative. I'm George Kafetsis, a master's graduate from Thomas Euler's lab, and currently a PhD student with Tom Baden. And as your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from University of California, Berkeley, the Paul Licht Distinguished Professor in Biological Sciences and member of the Helen Wills Neuroscience Institute there, and of course, member of the American Association for the Advancement of Science, Professor Marla Feller. Marla is a physicist by training, and after her bachelor at Berkeley, she studied surfaces and liquid crystal interfaces by means of second harmonic generation for her PhD with UN Ron Sen. I do not know what credit is due to the Woods Hole summer course on neural systems and behavior for Marla's passion in biophysics and neuroscience, but that started becoming evident through her postdoctoral years. First working with David Tang at Bell Laboratories and developing optical methods for studying the presynaptic terminals of the frog optic tectum, and then as a Miller postdoctoral fellow with Carla Sachs, using the Retina as a model system and doing seminal work on spontaneous activity. Marla started her lab at NIH in 1998, held the position of associate professor at the University of San Diego, and in 2007 she returned to Berkeley, where nowadays she is a professor at the Department of Molecular and Cell Biology. And before I move on to more research-specific introductions, I consider it highly important to mention that throughout the years, Marla has received many awards, not only for her research, but also mentorship. In her lab, they employ both optical and electrical techniques, and by focusing on mice and their retinal circuits, they study spontaneous activity, how it is generated, and what roles it plays in the development, organization, and maturation of neural circuits and systems. And today we will have the pleasure of hearing about their latest findings in her talk entitled Interplay Between Circuits that Mediate Spontaneous Retinal Waves and Early Light Responses during Retinal Development. So without any further ado from my side, please all welcome Professor Feller. Marla, the stage is officially all yours.
Great. Wow, what an introduction. Thank you, George. That was great. So let me share my screen. Okay, is it all working, George?
Yes, we are good to go.
Okay, let me just hide this. Great. Okay. Well, George, thank you so much for that introduction, for the invitation to participate in this series, and also to Tom for organizing the series. It was great during the pandemic. It kept us all sane. And I greatly appreciate that it's continuing to go on. So I shortened my title for today to make it a little bit more tractable. And what I'd like to do is talk to you about work that my lab focuses on, having to do with neural activity, how it's generated, and what role it plays in development at stages of development that are prior to the photoreceptors wiring in and vision begins. So just as an overview of what I'm going to talk about today, I want to start by showing this picture of the people who are in the lab. This is actually a picture from the lab a year ago, but it's going to be these people's work who I'm focusing on today. And so I'll be highlighting them as we go along. This is on a beautiful island that's in the middle of San Francisco Bay on a hike that our lab took. Okay, so let me just overview the sources of activity that are present prior to the maturation of vision. So the first we'll be talking about, as George introduced, are these spontaneously generated retinal waves and how they provide this robust source of depolarization with these very interesting propagation patterns that change across development. So I'm going to start by talking about that. I'm then going to talk about a second source of activity, which are generated by these intrinsically photosensitive retinal ganglion cells, which are functional very early in development and mediate light-guided behaviors. And then I'm going to introduce some strategies that the lab is using for disentangling the role that these two sources of activity play in visual system development. And maybe if there's time at the end to expand out a little bit on how the field can move forward, making clear that one has to disentangle these two sources of activity when thinking about development. Okay, so just to get everyone on the same page, I'll briefly introduce the retina. So the retina is this neural circuit that lies at the back of the eye. Light enters the retina and gets focused on. I'm sorry, enters the eye, and by the lens that's in the retina gets in the eye. Oh my God, gets focused on the retina that's at the back of the eye. Now, if we look at a cross section, the retina is not just a single plane of photodetectors, but it's actually contains many neural circuits. And I'll be talking a little bit about these circuits during development. So in the adult retina, light sort of passes through all the tissue. It activates the photoreceptors that are at the back of the eye. And then the output of the retina are these retinal ganglion cells, and it's their axons that make up the optic nerve and carry all the information about the visual scene to the brain. And that the processing that happens within the retina happens because of these interneurons that link the photoreceptors to the retinal ganglion cells. Now, the periods of development I'm going to be talking about are long before this circuitry is established. And to give you a sense of when that is in development, I'm showing you a schematic here of human brain development before birth. So the way that the visual system gets wired up is the eye actually develops independently of the brain. And at this incredibly early stage of development, retinal ganglion cell axons project to their targets in the brain. And humans actually open their eyes at 5 months gestation. So all of the work we do in our lab is in mice, and most of the work that I'll be talking about is sort of in this period of time that precedes the 5-month period right before eye opening. Okay, and so and at this time, clearly in humans and in mice, there isn't a lot of visual experience. And so there are other strategies that the nervous system is used to sort of activate these early circuits. So just to give you a sense of what the retina looks like at this period of development, this is again a schematic of the adult retina on the left that I showed you with ganglion cells at the bottom and photoreceptors at the top. Now, in the early stages before postnatal day 10, which we call P10, it turns out there are photoreceptors that are there that are postmitotic, but they have very small outer segments. And most importantly, those excitatory interneurons, the bipolar cells, have not formed their synapses that connect the photoreceptors to the ganglion cells. There are also amacrine cells that are present and starting to form synapses with the retinal ganglion cells. But you can essentially say the retinal ganglion cells are separated from any influence from the photoreceptors. So just because they're separated from photoreceptors doesn't mean that they're silent, but instead they're spontaneously generating activity. And this activity is what we call retinal waves. And so this is a movie actually that was taken by Alex Tidiak while he was a postdoc in the lab. And we'll be talking more about Alex's work in just a second. And what Alex did here is he isolated a mouse, a retina from a mouse that expresses GCAMP in ganglion cells, and took a movie of the spontaneous activity while focusing the 2-photon microscope on the ganglion cell layer. And this is what the activity looks like when he does that. And you can see that there's this spontaneous increases in calcium in these cells. And we know from a lot of work that these increases in calcium are due to these cells firing action potentials. So this is calcium that's coming through voltage-gated calcium channels and some neurotransmitter receptors, but it's all mediated by depolarization. So the lab is very interested when we want to understand the circuits that generate waves and what role they play in development. We actually look at these propagation properties of the waves of gaining insight into both of these questions. And so we have been really excited about using a new technology, a new what we call macroscope that was developed in the lab by Ben Smith. And this is just a very simple epifluorescent scope where we have a nice 4x objective, but and a really nice CCD camera. And Ben has designed an imaging system that really very nicely projects this information gathered from this objective onto the camera. And by optimizing the system, we have a field of view that's 5 millimeters across and with a resolution of 1.5 microns. So we have cellular resolution, but what this allows us to do is image these waves over the whole retina. So this is a schematic of sped up activity in the retina, and this macroscope is allowing us to image this activity really to gain much more insight into spatiotemporal properties. So this is important for a couple reasons. So as I was saying we care about these spatiotemporal properties because they give us insights into the circuits that mediate these waves and what role they might play during development. And this is interesting because waves actually persist for an incredibly long time during development. So during this period of development, the retina itself is undergoing a tremendous amount of development itself. So when we first see waves, which we see embryonically, the retina is made up of a lot of dividing cells and migrating cells, and the ganglion cells are mature and they've sent their axons. They've just reached their targets in the brain, but there isn't a lot of synaptic circuitry going on. From work that was done by Jeremy Kay's lab, we know that these Starburst amacrine cells which are going to be really featured in understanding circuits for retinal waves, are actually present, but they're just migrating into the position far from their adult morphology and positioning. Stage 2 waves are apparent later in development during the first postnatal week. And this is a circuit, at this point, we know that these Starburst amacrine cells have actually started to form synapses with each other and with retinal ganglion cells. And those synapses are cholinergic. So Starburst amacrine cells release acetylcholine. And we know during the stage of development that they themselves have nicotinic acetylcholine receptors, as do the ganglion cells. And so we think of this as a kind of cholinergic circuit. And then stage 3, which happens during the third postnatal week, become present, is when those bipolar cells finally form those synaptic connections with the ganglion cells and the amacrine cells. And at this point, the circuitry that mediate waves switches. So at stage 2, we can put on nicotinic acetylcholine receptor blockers, waves go away. We do that in stage 3, waves are still there. And instead, in order to block them, we have to use ionotropic glutamate receptors. I've actually included some references down here, which there are many, many papers by quite a few labs that have identified the circuits that mediate waves, but I like to highlight the ones that are kind of my favorites. There was a nice one by Kevin Ford from our lab in 2012. Aaron Blankenship studied stage 3 retinal waves in our lab. And actually, there's a really fantastic paper that I wish came from our lab. That came from, was done by a crew in Kirchensteiner, which is a really beautiful model that sort of has gotten at the mechanisms that underlie stage 3 waves. And I am going to highlight some work that is just coming out from our lab, should be published any minute, we hope, but right now it's in bioRxiv, and this is work that was led by Christian Buffo. Okay, so so the work that I'm going to be talking about was as I said, led by Christian Buffo with sorry, I'm not going to talk about that, and contributions from Maya, Andy Chen, who is an undergraduate in the lab, and Ben Smith. And this is now the macroscope imaging that we've done of these waves across these 3 stages of development. And so what you're looking at here is this beautiful slide that are macroscope images of stage 1, stage 2, and stage 3 waves. What's shown on top, I should say that the pink, the magenta background is just essentially an average. It's of all the activity that happens across this retina, and then hopefully in the green you can see the delta F over F signals, and you can see the difference in the imaging. What's shown in the top, what's shown at the bottom is the segmentation algorithm, but that Ben Smith has put together that really identifies where the waves are happening, and you can really see a lot of differences. So for stage 2, you can see there's this one mammoth wave that's slowly making its way across the entire retina, while in stage 3 you have a lot of littler waves that are actually propagating slightly more rapidly, and then the stage 1 waves seems to have a lot of what we think of as kind of false initiations, and every so often you get one of these big propagating waves. And so another way to sort of summarize what this activity looks like is in this nice representation that Ben has developed. So if you look at in this plane, this is an XT representation. This shows you the spatial extent of the waves that have propagated during about 2 minutes of recording. And then as it rotates and finalizes here, you can see what that looks like over time. So stage 2, we had one big wave that propagated across the retina. Stage 3, there's a lot more complexity in those waves. And stage 1 has a lot of little tiny initiations with some big larger propagating events. Okay, and our lab is super interested in doing this full characterization because and to trying to relate this to the circuits that mediate these waves, as well as what role they play in development. Okay, so let's focus a little bit on how are we going to get this question about what role do these waves play in development, and so let's talk about what's happening in development at this time. So I'm first going to talk about retinal projections to the brain, and this is where waves have been implicated quite clearly in playing a role in development. So during this early stage 1 waves is when retinal ganglion cells are actually innervating their primary targets. They've reached their targets in the brain, and in some cases are starting to innervate them. And then during this extended postnatal period, actually primarily during stage 2, there's a lot of refinement of those projections. So there's retinotopic refinement of retinal projections to the colliculus. There's eye-specific refinement of projections to the colliculus, and the lateral geniculate nucleus of the thalamus, and there's also laminar refinement in these target tissues as well. So the way that we get at what role waves play in driving this refinement is to do a loss of function experiment. And the one that has been very productive for us is a mouse that's missing a particular subunit of neuronal nicotinic receptors, the Beta2 subunit. So we use this mouse a lot. It has greatly reduced and altered retinal waves between P1 and P8. It's and it still has stage 3 waves. They come on a little bit earlier, but during this period, during the first postnatal week, there's very reduced waves. And so, so to show you how convincing this data is, that in this knockout, we have a strong phenotype. I'm going to turn to some experiments that were done by Alex Tidiak while he was a postdoc in the lab. Everyone should know that Alex now has his own lab at Vanderbilt University that's just getting started, where he's continuing to work on spontaneous activity in the visual system, but also in sensory motor systems and setting up circuits that mediate twitches. It's totally cool work. Everyone should check out his website. Okay, so what Alex did in the lab was to look at some experiments where he looked at eye-specific segregation. And the way he did these experiments is he did injections of cholera toxin and anterograde tracer, where he put green in one eye and pink in the other. And these get anterogradely transported, and they allow you to look at what their projections are like in their targets. He then used this method for clearing the brain so that we could look at retinal projections throughout the brain. And when he does these experiments, you can these are now what you're looking at is a projection through the lateral geniculate nucleus. You can see that the contralateral projecting axons take up a large part of the projection to the LGN, while the IPSI is a smaller projection, which just showing you one section in the middle. I mean, you can see that there is this segregation. So this segregation actually comes about by postnatal day 8. It's present by postnatal day 8. And there's a lot of overlap between these projections at postnatal day 4. And so there is this segregation process. And so when we repeat these experiments in the Beta2 knockout, you see that this segregation has really failed. And it's based on these experiments and experiments like this that we've been able to show that these retinal waves during this early period of development are very important for refinement of retinal projections to their targets in the brain. Okay, but now I'd like to focus on work that the lab has done to understand the role for waves, perhaps in retinal development. And so what's happening during retinal development during these ages? Well, during stage 1 waves, there's a tremendous amount of neurogenesis and positioning of cell bodies. And then there's also there's a couple periods of cell death, but the one I want to focus on is in this postnatally between P1 and P4, a lot of ganglion cells undergo cell death, with estimates of high as 50% of ganglion cells will die during that time. And so in order to sort of get at these questions, we need to come up with a way of manipulating stage 1 waves. And we already have a way for manipulating these stage 2 waves during cell death. Okay, so now I want to talk about this work that was the focus of Christian's thesis project. I would say this project was really, Alex was really kind of the mentor who guided this project along. And there were significant contributions from Andy Chen. Andy was an undergraduate in the lab at the time, and he's now getting his PhD at Harvard. And of course, we have the contributions from Ben on this study. Okay, so what this team did is they did 2-photon calcium imaging, and what you're looking at here are different cells, individual cells, as a function of time, and these are these kind of raster plots that can tell you what their participation in waves are. So this is what the control looks like, and you can see waves are happening around once a minute. And what they found by doing these experiments is that stage 1 waves were significantly inhibited by a gap junction antagonist called MFA. And that was something that was sort of known from previous works from our lab and other labs. But a bigger surprise came when we found that these waves were actually significantly inhibited by a nicotinic acetylcholine receptor antagonist. That despite the fact at this incredibly immature state of development, where I had mentioned the Starburst amacrine cells are far from their target position in the INL and in the ganglion cell layer, they are still releasing acetylcholine, and that acetylcholine is contributing to waves. So now the fact that we know acetylcholine is involved in mediating these waves, we look to the Beta2 knockout to see what these stage 1 waves look like. And what we saw is summarized here. So we see that unlike at stage 2, we still see a lot of wave activity in the Beta2 knockout. But interestingly, that remaining activity is not blocked by nicotinic acetylcholine receptors and is completely blocked by a gap junction antagonist. So the waves are still there, but there has been this, I guess we could say, compensation so that the remaining gap junctions are sufficient to propagate these waves. But what you can tell from this is that the waves are actually greatly reduced. So if you were to look at the percent of ROIs that are active during waves, we can see that in some of the big waves in control, you can get 50 to 70% of the retina participates in those waves, but this is significantly reduced. So that's another way of saying the waves are much smaller in Beta2 knockouts, and what's not shown here is they actually propagate at half the propagation speed. So waves are reduced in the Beta2 knockout during stage 1, and they're significantly reduced and almost absent in stage 2. So what does that do in terms of retinal development? So to get at this question about cell death and cell positioning, we turn to a particular ganglion cell type that we have a good marker for, and that is these intrinsically photosensitive ganglion cells. I'm going to talk about them a lot in just a moment, but just suffice it to say this is a particular cell class that is present subtype of retinal ganglion cells. And this is actually the work that was done by Andy Chen. And so if you look at all of the labeled cells in an OPN4 Cree crossed with the TD tomato P1 and compare it to postnatal day 7, you can see there's a dramatic decrease in the density, although the retina has grown a little bit. And if you look at these at higher magnification, and I want to emphasize again, these are all images taken from the macroscope, including these high resolution images, you can see that there is this reduction in density. And then if you compare this between the wild type and the Beta2 knockout, you don't see a significant difference. So this was quantified in the following way. First, if you look at the number of these cells, the density of cells at postnatal day 1 to postnatal day 7 in across development, you see that there is a dramatic decrease, and this is, we know, is due to cell death. And if you now compare this in the Beta2 knockout, you don't see that there's a significant difference in the density of cells at postnatal day 1. Nor do you see, and you still see a dramatic amount of cell death that happens in the first postnatal day week. So it seems that the absence of waves during stage 2 has not prevented cell death from happening. We also did a couple measures of the organization of the somas, what the retina world likes to refer to as the Mosaic organization. And here we plotted the nearest neighbor distances in a cumulative histogram plot. So postnatal day 1, you can see that there's a lot of overlap between what we see in wild type and Beta2 knockout, and that there is a significant increase in the nearest neighbor distances that you can see between the wild type and the Beta2 knockout. As a way of talking about the organization of the somas within the retina, we calculated the regularity index, and that's the nearest neighbor distance as a function divided by the standard deviation. The higher the regularity index, the more regular the mosaic is, and the lower it is, it's closer to random. And so we found that actually the regularity index did not change, and actually this value of being around 3 is what is predicted for a simulation of randomly distributed somas. So the somas remain randomly distributed, and there's no change in that in the presence of in the absence of normal retinal waves. Okay, so we don't think that retinal waves are playing a role in these important developmental processes, although of course a more significant blockade of stage 1 waves are necessary to completely rule that out. I now want to very briefly talk about experiments that just were published last year from the lab implicating stage 2 retinal waves in the assembly of a retinal synaptic circuit, and one that is very dear to our hearts, and that is the direction selectivity circuit. So this is a paper that was done by team direction selectivity that includes again Alex, Karina Bystrong, who's a graduate student in the lab, Josh Tworig, and Maya Pitcher. Okay, so what did we find? So I'm not going to be introducing direction selective cells, but for those of you out there who know that there is a population of cells within the retina that prefer one direction of motion over another. If you record with 2-photon calcium imaging over all the direction selective cells in a field of view, you'll find that those preferred directions cluster along 4 axes. So you'll have some cells that prefer left, right, and then some that prefer up and some that prefer down. Those cells are all present right when the eyes open, so you don't need any visual experience for the establishment of this direction selectivity. And when they looked at the Beta2 knockout, which just to be clear is lacking waves between P1 and P8, they have this remarkable result, which is there seems to be no horizontal direction selectivity in these mice. So this lack of waves during this early stage of development prevented the development of horizontal direction selective cells. So those cells are still there. They just have lost, and they're still responsive to light, but they have just lost their directional preference. So that indicates a very specific circuit has been disrupted. The robustness of this phenotype, or the implications of this phenotype, were actually described by J.C. Kang's lab over 10 years ago, 15 years ago, oh my goodness, when they showed that mice that these Beta2 knockout mice actually lack a horizontal optokinetic reflex, a behavior that's known to be mediated by direction selectivity, but their vertical reflex seems to be present. So something about waves is very important for setting up the circuit, and sort of getting at that question is a major focus of the lab that I won't be talking about in this talk. Okay, so as an interim study about retinal waves, I just want to say again, retinal waves are this robust source of activity before vision, and they're required for the precise connectivity to emerge throughout visual the system for some circuits within the retina and not others, and of course for retinal projections to their targets. Different stages of waves have different spatiotemporal properties, and that retinal activity is implicated in the wiring of direction selective circuits, but not in the cell death, at least of IPRGCs. Okay, and now when thinking about these questions, and we'll get to this at the end, I just want to say when we talk about whether or not retinal waves are involved in a particular developmental process, we can think about it either at the level of, do the spatiotemporal properties of those waves matter for that developmental process, or is it just the fact that there's some cell autonomous activity in each cell that drives it? And that's a big question that we're always asking ourselves when we're looking at how retinal waves affect development. All right, so now I want to turn to this other topic, and I have to say I have been studying retinal waves since I was a postdoc for a very long time. I had said forever that they are the only source of activity before vision is established. And then this paper came out, Juliet Johnson and David Copenhagen, where they showed that mice pups that are only 6 days old will actually turn away from the light. So this is a behavior experiment. We have 5 different mice. There's an LED to the left. And at some point, these mice are the light's gonna flash. You can see all these mice are turning away from the light. Okay, so I told you that this is not photoreceptor mediated, rod and cone mediated, because those photoreceptors are not connected to the ganglion cells and therefore not connected to the brain. And so what mediates this behavior is actually a class of these retinal ganglion cells are these intrinsically photosensitive retinal ganglion cells. So if you create a transgenic mouse that doesn't have melanopsin, this behavior goes away in mouse pups. So this work from the Copenhagen lab really established that these cells are there and they're driving they're driving this behavior. Okay, so IPRGCs are present and they have been implicated in many developmental processes. And every time I open a journal, there seems to be another process that they've been implicated in. So just to give you a sense of the development of them, IPRGCs are light sensitive as early as E15, and the different types of IPRGCs are fully differentiated by postnatal day 4 or 5. They have innervated the suprachiasmatic nucleus in the LGN by postnatal day 0. They have been implicated in some retinal development in work from Jordan Renna's lab, excuse me, that having to do with the lamination of cone somas because of a transient projection that IPRGCs send up to the developing photoreceptor layer. They've been implicated in the maturation of vasculature, either by suppressing angiogenesis or regression of a developmental vasculature called the hyaloid. They have been implicated in the maturation of circadian-driven functions. And there was a really interesting study amongst quite a few that they're very involved, that this early light activation of IPRGCs is critical for synaptogenesis in cortex via modulating release of oxytocin during this very important early developmental period. So there's been a tremendous amount of work, right, that says that IPRGC mediated light responses are implicated in lots of development that's going on at this time. But there is this question, and this is actually was the focus of Franklin Cavall Holmes' thesis, which was, how does a mouse tell the difference between a wave and light? So as far as we knew, all ganglion cells participated in waves, and therefore all IPRGCs participated in waves. And so how would a mouse know whether or not an IPRGC is depolarized by a wave versus whether it's stimulated by light? So this is a question that Franklin was really focused on. Franklin graduated. This is his graduation party during the pandemic. Which was in 2020. Franklin is currently a postdoc in Michael Doe's lab, where he continues to study IPRGCs, somehow only in the adult. I don't know, but I'm sure it's very interesting. I'm sure he's doing fantastic work there. Okay, so I just want to very briefly go over Franklin's work. His thesis work was published in 2 papers whose references are shown there. And I just want to sort of show you what his experiments look like. So what Franklin did is he would do a retina dissection. He would put his retina under one arm of this 2-photon microscope in order to do calcium imaging. And then he used a projector system that would project blue light in order to activate these cells. And just to give you an idea of what this data looked like, you can see on the right that when there are these flashes of light, of blue light, that you can very clearly see light responses from IPRGCs. Now, in these particular experiments, he's blocked waves in order to isolate these IPRGC responses. And when he did that, for some of you who are looking, you could see lots of cells that were responding, but they actually seem to respond with very different kinetics. And so what Franklin did is he using the strategies that were developed in the Euler lab by Tom Baden and Behrens to use a sparse principal component analysis to classify these cells into functional groups. And he found that there were about 6 functional groups. And you can see that, and if you look at the average response, and so what you're looking at here are the responses of cells to long duration stimuli that are increasing intensity. And you can see some of these cells have a very low threshold. So they respond to very low levels of light and they have a sustained response, while some say have a higher threshold and have a transient response. Franklin went on to try to assign these functional groups to anatomical groups. And just to introduce this to people, this is a summary plot that talks about the multiple types of IPRGCs. There's 6 different types that have been identified. Tiffany Schmidt's lab has shown that many of these are present very early in development. And so that the different subtypes are identified by transcription factors and also where their projections are in the brain. And so when Franklin would map his functional groups onto anatomical groups, there, he had 6 functional groups or 6 anatomical groups, but what he found was that they did not map precisely. So for the first functional group, those mapped very well onto the M1 IPRGCs. And so there was a functional group that mapped onto an anatomical group, but all the other anatomical groups actually were mixed into forming these functional groups. And that mixing of these groups happened because Franklin showed there was extensive gap junction coupling between them. Okay, and to give you a sense on how powerful this gap junctions are in terms of mediating the responses of IPRGCs, what you're looking at here is the impact of a gap junction blocker on the light responses of the different functional groups. So if you look at the response in control conditions, you can get to a light stimulus, you get a fractional change in fluorescence that varies. And if you compare that to the responses in the presence of MFA, almost all the cells, all the functional groups lose their light response except the M1s, right? So the M1s are able to keep their light response even when gap junctions are blocked. Okay, so what does this mean? So remember, we want to get at this question about how the animal tells the difference between a wave and between a light stimulus. And so the first thing we needed to, so now we want to get at that question to figure out what type of IPRGCs might be mediating photoaversion. So we have one class, the M1s, that are kind of cell autonomous. Then we have the other class that are all gap junction coupled together. And so Franklin's strategy to get at this question was to take advantage of many of the tools that have been developed to sort of do loss of function in these 2 groups. So we know that transduction in the M1s are mediated by TRPC6 and TRPC7 ion channels, while they have a weaker contribution to the other functional types, and that we know that if you can do intraocular injections of gap junction antagonists, that will allow you to block the effects of the other cell types. Okay, so we took advantage of this triple knockout that was developed by Lutz Birnbaumer and was given to us by Tiffany Schmidt, who is a collaborator on this project. So what Franklin did was the following: to quantify photoaversion is he mapped the head. He literally put an arrow on the head of each of these little mice, and then when they were exposed to the light, he looked at their head position, and eventually they turned their heads at some distance away from the light. And when he did this in control, he looked at how much maximum head angle there was in the dark versus in the light, and he used 2 different intensities of light. He found that in the C6-7 double knockout, that actually that photoaversion was completely prevented at the medium light intensities, and there was a little bit of photoaversion at the highest light intensities. However, when he blocked gap junctions, he did not impact photoaversion in these cells. So intraocular injections of gap junction antagonists did not prevent photoaversion, really sort of arguing that it seems like it might be M1s. But to really establish that, he made use of this incredible mouse line that was developed by Samer Hattar's lab, where it's an OPN Cree that's crossed with this brain 3B and DTA. All you need to know is it kills all of the IPRGCs except for this one subset of M1s, which are brain 3B negative, which project to the suprachiasmatic nucleus. All right, and when he killed every IPRGC except for these M1s, what he found is that photoaversion was completely normal. And so I think with these experiments, we can safely say that the M1 IPRGCs mediate photoaversion in these pups. Okay, so now we're ready to answer our question. How does the mouse tell the difference between sensory experience that we know is mediated by M1s, and then, of course, this spontaneous activity that's mediated by the retinal waves? And so these experiments, so what Franklin did in collaboration with Andy Chen, did the following experiments. He looked at cells that participate, where he looked at waves, and then he also looked at light responses. So if you look at, these are delta F over F transients from 3 different cells that are non-IPRGCs, and where you see these red dotted lines is when there's a wave, and you can see that these cells all participate in retinal waves, but none of them respond to the blue light. He then did experiments in where he did the same experiments, but now he looked for the putative IPRGCs, and those were because they had light responses, and I'm sorry, they were GFP positive, right? Because we were using a mouse line that had GFP and the IPRGCs, and you can see that they had light responses, and some of them participate in waves and have light responses. And then it turns out there was a subset of these cells, which were the M1s, which have this very distinct light response, but they actually don't participate in waves. That M1 IPRGCs appear to have a much smaller participation in waves. So in analyzing this data, if we look at, say, one of the different functional classes, and you do a wave-triggered average, you can see that you get a robust response in IPRGCs. But if you look at the M1s, there's no consistent response. And Franklin did this for a variety of different light stimuli, and you can see that if you look at the delta F over F in terms of their response to light versus how strongly they're depolarized by waves, you can see that M1s are much more strongly depolarized by waves than they are by light. So that seems to argue that M1 IPRGCs represent an isolated channel for mediating photoaversion during development. Okay, so to summarize the second part of the talk, light stimulation of IPRGCs mediates behavioral responses at the same age as their retinal waves. The visual system differentiates spontaneous from light-evoked events by exclusion of M1 IPRGCs from waves, and that the light stimulation of IPRGCs, oh, sorry, I just repeated something. Sorry. Okay, so forget that third one. And that the visual, oh, I repeated them all, because they're really important findings. Okay, so this question about how M1 IPRGCs are excluded from retinal waves is actually something that I would really love to follow up. So it could be that M1s don't have nicotinic acetylcholine receptors, and that's why they don't participate in waves. And certainly the single-cell RNA-seq data seems to argue that they have a lower expression of nicotinic acetylcholine receptors, or it could be that they're strongly depolarized by waves and they go into a depolarizing block. And Franklin tells me that he's doing those experiments. I'm trying to convince Franklin to do these experiments while he's in Michael's lab. Michael, please let him do these experiments, but hopefully we'll have an answer for you for that very soon. Okay, so now I want to take a few minutes to sort of get at this question about the interplay, other experiments the lab has done to get at this interplay between IPRGCs and waves, because they're both going on at the same time. Okay, we know that waves depolarize IPRGCs. We know that light does not dramatically influence stage 1 or stage 2 retinal waves. So when we do calcium imaging, we see no difference with how light affects the spatiotemporal properties of retinal waves. Jordan Renna found by doing MEA recordings that when you turn on the light, that there are some cells that you increase the burst duration of some cells during when you turn on the lights during stage 2 waves. So, but the frequency of the waves and the propagation speed of the waves doesn't change in light. If we acutely block retinal waves for about an hour, or if we look in the Beta2 knockout mouse, we have a dramatic increase in the number of light-sensitive cells. We'll get twice as many light-sensitive cells when we've blocked waves for this sustained period of time. And we'd love to understand how that happens. So our current thinking is that this is due to an increase in the gap junction coupling, right, between IPRGCs and perhaps non-IPRGCs. But we're also open to the possibility that this could lead to some sort of changes in melanopsin expression that changes the sensitivity of different IPRGCs. And that's an ongoing area of research. And we're really interested in this question about whether extensive coupling of IPRGCs contribute to retinal waves. So they are a very gap junction coupled network. We know that gap junctions, particularly during stage 1, are very important for retinal waves. And we wonder if that coupling is what's critical for plays a role in these waves. There were some experiments done by Samar Hattar's lab that showed when you killed IPRGCs, that had an impact on waves and eye-specific segregation. And so that's kind of supportive of that particular model. And now I just want to expand out a little bit more about how this early activity influences development. We are not the only lab studying this, and there's a tremendous number of labs. I just kind of want to highlight some interesting findings that are out there, which is that this early retinal activity is known to regulate the timing of corticothalamic innervation. So when cortex innervates the lateral geniculate nucleus, that's something whose timing has been identified as being important for the timing of which is controlled by retinal waves. That there was an amazing paper that came from the Lopez Bendito lab that showed that the segregation of visual and somatosensory circuits within the superior colliculus is something that's dependent on actually early waves, perhaps stage 1 retinal waves. And that this idea that spontaneous activity is important for development of retinal microcircuits has proven to be the case in actually the Drosophila system. So there's been these wonderful studies by Aiken while he was in Larry Sapersky's lab, and now in his own lab at UCLA, that shows that there's spontaneous activity in the Drosophila visual system, and that blocking that activity also prevents the development of some specific circuits within that visual system. Our lab and others have really focused on the interaction between retinal waves and glia. So we know that work done by Julie Rosa while she was a postdoc in the lab, and Josh Twareg, who's was a graduate student in the lab and is now graduated and is now staying on a little bit longer as a postdoc, has shown there's robust signaling between retinal waves and glia. Retinal waves cause transients in the glial cells, and we're really interested in what role this neuronal glial signaling plays in development. And I should say this was also seen in zebrafish in work done by the DO lab. There have been these amazing talks on this very series that I have been watching that have gotten us very interested in and whether or not retinal waves and signaling via the glia might be involved in the development of the vasculature. These were talks by, excuse me, that we heard here in the series, sorry, and we're carrying out some experiments by a new graduate student in the lab, Samira, who's going to be sort of getting at this question about whether blocking retinal waves will have some effect on development of the vasculature. Another sort of thing that we're extremely interested in is that retinal waves, our lab has been able to show with imaging using a cell-based fluorescence sniffer technology that retinal waves drive release of dopamine. And we're really, but we also know that in the adult, certainly that activation of IPRGCs drives release of dopamine. And Jordan Renna has some very good evidence that this also might be important for development of the retina itself. And so we are continuing to do experiments to develop methods for looking at live imaging of dopamine in the retina, and these experiments are being carried out by a new postdoc in the lab, Rongshan. And then there could be all these things we haven't thought of yet, right? So there could be a lot of developmental processes that are going on. And as I had said earlier, I'm really interested in this idea of cell autonomous effects, like do these periodic depolarizations or the strong activation of light do something at the transcriptional level in cells. And so I'm really excited by a collaboration that we're doing with Karthik Shekar, who is a new faculty member here at UC Berkeley and has really novel approaches for studying single cell RNA-seq data and using that as a way of looking at the development of different ganglion cell types. And retinal types. And this is going to be work that is being led by Rachana, another new postdoc in the lab. Okay, so this is the lab. We love retinal waves. And this is the current state of the lab. The current members of the lab, as I mentioned, Josh has recently graduated, staying on a little bit longer to finish some exciting experiments he's doing on the development of direction selective circuits. I want to call out to the recent graduates. I think I've told Alex is a professor at Vanderbilt. Oh, Christian is now starting a really exciting fellowship at the World Bank. Franklin is a postdoc in Michael Doe's lab. Andy Chen is, I think, rotating in Michael Doe's lab. And Yizhen Zhang, who worked with Franklin on his projects, is now a PhD student at Hopkins. And for those of you who know other recent Feller Labs graduate, Matthew Summers recently graduated, is now at the Allen Brain Institute, and Malik El-Kesni is at the Institute Photonics in Barcelona. All right. Thank you very much.
Thank you very much for this very impressive talk, Marla, and your meticulous attempts in trying to figure out what it might be, including something we haven't thought about just yet. So let me go ahead and post the Zoom room link in case people want to start joining us already, and remind to our audience that after this first initial moderation from my side, we will be continuing in a more informal Zoom get-together fashion. So please make sure to follow the link in case you want to be part of the discussion later on. So there are already questions appearing in the chat. Before I go into them, one very naive, borderline stupid question that I have is, so when it comes to studying retinal waves, do we know if there's a circadian component in them?
Now, that is not a stupid question. And, okay, I'm going to take a step back to answer this question, George. It's so weird to be in a Zoom room because as far as I can tell, I'm only talking to George. And so, so I'm going to tell George that as he mentioned, I was a physicist, and people often ask me, what is the thing that I miss about physics? And I have to say, the one thing that is frustrating about neurobiology is there's so much variance, and we don't know what the variance is due to. And oh my God, all our variance could be due to circadian rhythm, right? So it is something that certain people in the lab have tried. Like if you do retinal wave imaging in the middle of the night versus, okay, no one's done it in the middle of the night, but morning versus evening, we don't see big differences. But I would say that we have not studied that rigorously enough to really make that conclusion. I will say when we put on dopamine receptor antagonists, in wild type retina, we don't see an effect on waves. So if the circadian rhythms are changing dopamine levels, then that's not likely to be a method. But we do all of our experiments in C57s. They don't have melatonin, and so it could be that we're not studying the right mouse to figure that out. So yeah, it might be important. We haven't looked at it.
All right, thank you very much. So following your comment, people are already joining us in the Zoom room. So I would like to ask them to keep their cameras and their microphones muted for the time being as I moderate the conversation with the questions that appeared in the chat. So the first one is from Daniel Kirschensteiner. Great talk. Since M1 IPRGCs do not participate in waves, do they differ in their eye-specific segregation and retinotopic refinement from other RGC types?
It's a great question. We have not looked. We have not looked at that. I think that would be a really fun thing to address now that we know that they seem to be doing this on their own, right? To be independent of waves. I think I would love to look at that.
The next one appearing in the chat is from Gautam Awatramani. Fantastic talk. I was wondering, are Starburst amacrine cells the source of acetylcholine for stage 1 waves?
So we haven't tested that explicitly. If looking at Jeremy Kay's work, actually, I guess he didn't. I don't know if Jeremy, if you're out there, you can answer in the chat. But I don't think he did like choline acetyltransferase staining or anything. He just had a marker for Starburst cells. So our assumption is that they are. I know that there have been some reports that maybe horizontal cells transiently express acetylcholine during development. So we haven't tested explicitly whether it's Starburst cells, but that's our assumption.
And the next one appearing in the chat is from Chase Helmer. Is there any possible contribution of nicotinic acetylcholine receptors in bipolar cells to stage 3 wave propagation, like propagation speed?
Yeah, I mean, that was Chase with your studies revealing that those existed have made us realize that that's something we have to consider, and we haven't looked at that explicitly, but absolutely. We don't see, I guess I will say during stage 3 waves, we do put on nicotinic acetylcholine receptor blockers, and we don't see an effect on the propagation properties of waves, but we haven't done that sort of at the level of the macroscope imaging that we're doing now. We've done it more on like smaller fields of view, and so we don't see a big effect on frequency, but there may be other like propagation speed and stuff like that might be important for us to look at. So it could be a source of modulation. We do know that Starburst cells still participate in waves, right, during stage 3. So acetylcholine is certainly being released. They're just not contributing, as far as we can tell. They're not the major source of propagating retinal waves.
Right, and given that there are a lot, like you don't have YouTube open, so there are a lot of messages saying both thank you and greetings in the beginning. And I would like to thank the audience for, either waking up.
Everyone come into the Zoom room briefly. I want to say
or
hello.
Staying up late. I think I didn't miss any question that appeared already. So one of mine, like before, like if there's no more questions, I will be terminating the broadcast after this. So one of mine is like, so you mentioned that at stage 1, something that differs in terms of the patterns of retinal waves is that we have like a lot of failed initiations. Is it really failed initiations, or could it be like a more local function of these retinal waves, like shaping the circuits at a local scale and not global?
Yeah, I mean, I think we make that stuff up, right? So I would say, yes, failed initiation is something we made up. It could be that it's a little gap junction coupled network. It could be it's a little, source of spontaneous depolarization. We don't know what it's necessarily due to. So perhaps failed initiation is not exactly the right thing to say. I mean, one of the things that Ben has pointed out as he and Maya are sort of really spending a lot of time looking at the spatiotemporal properties across development are the transitions. And so stage 1 waves, as they convert to stage 2 waves, you see more of the big propagating events and few of those very small events that don't go anywhere, right? So there seems to be that gradual transition. And the same thing sort of happens when you go from stage 2 to stage 3. Now you don't have as many big waves, and they start having more frequent initiations, and they start to break up a little bit more. But then every once in a while, you'll have one of these big, what we think of as cholinergic waves. So they're really, so everything is kind of a continuum, and maybe these 3 periods are artificial in some sense, particularly between stage 1 and stage 2, and more work will have to be done to really identify that.
All right, thank you very much. One more question appeared in the chat, and I'm sorry if I'm mispronouncing the name, is from Amiroshan Deghani. Amazing work. Is the retinal waves causing the retinal formation and retinotopic organization, or is it more like a correlation? If it's causation, is it explained by Hebbian learning?
Yeah, so, all right. Is it causation? I don't know. Here, I'm going to wear my physicist hat. We don't ever prove anything in biology, so everything's correlation. And I will say we have the loss of function studies. Mike Crero has done a really very nice gain of function studies where he's done things where he's restored activity back into the Beta2 knockout and shown that if you restore activity, it has slightly different altered propagation properties, and he can show that some maps are sort of formed normally and some aren't. And he's also done some really nice experiments where he's used channelrhodopsin to sort of stimulate like the left eye and the right eye synchronously. So you have left eye versus right eye. And he's been able to show that if you do that, you can rescue eye-specific segregation as long as those left eye, right eye stimuli aren't coming too frequently. I think he found it had to be like a second or so different. I apologize, I don't remember that. And so that kind of does argue that the salient feature of retinal waves for driving eye-specific segregation is that there's a very small likelihood that the 2 eyes are ever firing synchronously in the same retinotopic location. And so I think that that's probably pretty safe to say. What mediates it on the other end, I think it's a huge topic of what many people have been studying. There certainly seems to be, you need synaptic transmission in order for it to occur, but it's not Hebbian in the sense that a classic Hebbian model, because IPSI is always so much weaker. So the model seems to be if IPSI is active, it will form that, it will be able to innervate that little IPSI patch. But it doesn't take over the whole retina if contra is silent. And so there seems to be like, you need activity to compete within that little patch. And so, it's not a pure Hebbian process.
Right. Thank you very much for the questions that appear. Thank you very much, Marla, both for the talk and for addressing them. And with this general end, whether it's like Hebbian learning or not, I think I will be, I will be I will be terminating the broadcast. I'm posting once again the Zoom room link in case people haven't had the chance to save it before, so they can join us. And given that we are already more than 10 people who probably have like questions for you, let's continue offline. Thank you very much.
David, Daniel, all my favorite people.